Hello, today I'm going to be talking to you about DNA extraction. So in simple terms, it is exactly what it sounds like. Extraction is just extracting DNA. Where are you extracting it from? Like, where are we taking it from? We're just taking it from the cell. So we have these cells, and inside your cell you have the nucleus. And your nucleus, you have chromosomes, and these chromosomes contain DNA, right? Genetic information, just, like the code that makes us us. It's like computer code, but for people, right? We're extracting it because it's inside your cell and it's inside the nucleus. And so we need to bust open or lyse the cell, just meaning we're opening the nucleus, so that we can have this genetic information that is contained here, kind of in a container, if you will, like we're opening the container by lysing or extracting the DNA. Okay, so there's 3 types of DNA extraction methods. They're all different. So we're going to start with organic or phenochloroform extraction. It has double-stranded DNA, which means that it has both strands, right? And then it is the oldest. So it's the oldest and the longest one. It has the most contamination. It has the most DNA loss. And to be honest, it's not that good for you. These chemicals here are carcinogenic, which means they cause cancer. They're very bad for you. So this is it's very old, so it's like the original one. You have your sample, right? So you put your blood stain in here. You add these chemicals. These chemicals are the things that are going to lyse or open your cell. So you're going to incubate it. So you have it at this temperature. You centrifuge it, which is kind of like spinning it. Then you add your chemicals in. You vortex it, and there's just going to be so many steps, which increases your chance of contamination because you're doing multiple things. And you'll see later that the other ones are significantly shorter. And then because there's more contamination and there's more steps, you're more likely to lose your DNA just because there's more places you could lose it in. All right, then we have Helix, which is shorter, as you can see, and significantly safer. This is going to give you single-stranded DNA. The first one would give you double-stranded. So you're going to have, let me see if I can do this, single strand instead of double stranded here. And you can see, so you add your sample and you add water and you incubate and you centrifuge it, and then you add your Helix beads, and and then you incubate it again, you centrifuge. But this is going to leave your sample with a bunch of junk, so it's going to be full of stuff, which can cause problems down the road, right? Because it's full of stuff, it can block other things. Finally, we have FTA paper, which unfortunately is only limited to knowns. Knowns are something you already know what it is, right? If you sample your DNA, you know it's your DNA. This can't be like the other two, which you have your unknown sample, right? So at a crime scene, if you collect a sample, you don't know whose it is. That's why you're collecting it. And so these two are going to be for unknowns or question samples. This is only for known samples. So it's the shortest because this FTA paper, once you apply your blood stain here, it lyses or opens the cells for you. It busts open the cells, if you will. And then you can just punch out a sample and place it in, and then you just remove the supernatant and then you add your PCR reagents. So it's short, quick, and easy. Last but certainly not least is going to be what is used oftentimes today for other question samples. Is going to be solid phase extraction. So there's different methods for this, but regardless, you're going to start by lysing your cells, which is just opening your cells. Then you're going to have something that binds DNA, generally magnetic beads. That's why there's a little magnet here. And these beads have an affinity or a liking for DNA. So they're going to be like, I want to hold this DNA. But at some point you want to let it go. So this is showing you have an elution or an elute buffer, and it's going to make the beads be like, I don't want to hold this DNA anymore. It's going to let it go. And then you're going to have your DNA. And finally, last but not least, these are my figures cited from each side.